morning, children. I feel extremely happy to welcome you all for this online learning. There's a proverb saying, hard work never fails. And another proverb states, there is no substitute for hard work. Every work of yours will bring you happiness at the later half. Keep working hard, prepare your lessons thoroughly, and gain knowledge every day, every minute. That is more important in your life. I'm very happy to share with you a good news that your seniors all have passed their 12th exam and they've come out with colorful colors. And I also wish you to get good marks and get passed through your 12th standard exams. As I've said before, we have completed our 4 lessons and we have stepped into our first lesson. And last week, we have discussed about the first part of the plant tissue culture. And in today's class, we'll be learning about the second half. Types of plant tissue culture, organ culture, second is meristem culture, third is cell suspension culture, and fourth one is protoplast culture. So these are the various types of culture which we'll be discussing in our class today. And we'll also learn about the applications of plant tissue culture and various other techniques in how which we can cultivate the plants. That is the protocol for banana cultivation and protocol for virus-free plants and disease-resistant plants, cryopreservation. So all these topics we'll be discussing in our today's class. Hope you will enjoy the class, gain knowledge and keep learning. That is more important. Come children, we'll learn the lesson.
We have been discussing about the various things that happens in plant tissue culture. In the first picture you can find a plant and an explant has been taken, a part of the tissue has been taken and it is kept in the culture medium to allow it to grow into a calyx. That tissue which has been transformed into undifferentiated mass of cells called the calyx, which is then transferred to a conical flask with a nutrient medium, and where you can find this calyx has turned to plantlets. In the test tube you can find small plantlet grown. This is what happens in a plant tissue culture, and once the plant is grown, we transfer that plant to a ordering process, that is planting it in the soil and exposing it to the sunlight. So this is what we discussed in our last class, and in our today's class we shall look into various other concepts, various other techniques related to plant tissue culture in our today's class. Hope you all have learned your previous lesson and ready for the today learning. We shall start with the various types of plant tissue culture. Based on the explant, some other plant tissue culture types are culture organ, meristem culture, protoplast culture and cell culture. In organ culture, instead of growing an entire plant, I concentrate only in developing certain organ which is of great need. So I concentrate only in culturing the particular organ. And in meristem culture, I concentrate only in the meristematic region, that is the region which undergoes repeated cell division. Similarly, in protoplast culture, I stick to my protoplast alone. I fuse my protoplast and then grow it into a new variety. And in cell culture, again, we are not growing an entire plant but a particular thing. So these are the various types of plant tissue culture which we will discuss in our lesson today. Let's start with our organ culture. This organ culture is very much specific for the culture of the embryos, anthers, ovaries, roots, shoots or other organ of the plant. Only a particular organ we are going to culture it. Okay, let's look into the picture and let's understand what happens in the organ culture. You have an isolated cell, you got a plant. From the plant, the leaf is taken up, we call it as the explant. From the explant, we remove the cell, we keep it in a culture medium. Cell is cultured in the culture medium, and in a few days, you will find raising of plantlets. The third picture, plantlets have been grown, and that plantlet is isolated and kept in a separate container. After a certain level of growth attainment, I transfer the plantlet to the hardening process. You can see in the picture, the last picture, where the plant is exposed to the sand, mineral, natural environment. So this is how the culture is formed. You may think the organs are not there, but the entire plant is formed. Yes, but in the entire plant we are specifying only to the organs. We are considered with the organ development and we isolate those organs from the culture. So this is all about your organ culture. The same kind of procedure, but the main intention in this culture is isolation or the production of embryos, anthers, ovaries, roots, shoots, or any other desirable parts that we require for our daily commercial use. The second type of culture would be meristem culture. In this, the culture of any plant meristematic tissues on the culture medium. You can look into the picture. The older leaves are removed by cutting or corneation. The young tips are taken up. We can find the meristematic tissues there. And this meristematic shoot tip with growing tips after removal of enclosing leaves, okay, the tip where the growth takes place, that is taken up for the culture. That tissue is kept in a paper, just wrapped and put it into a culture medium and closed with a cotton plug. After some times we find calyx are formed. Calyx are nothing but undifferentiated mass of cells. This calyx develops into small plantlets, and once they attain certain maturity, we transfer them for the hardening process, that is in the growing in the real environmental condition. So this is what is called meristematic cell culture. So only the growing tips are taken up. The cell division activated center points are taken and it is cultured to form the new plant. This is a second type of culture. First we discussed about the organ culture. The second we have been discussing about the meristem culture. Now let's move on to the next topic, that is protoplasmic culture. This protoplasmic culture is of high commercial values, which we can see in our next topic, that is protoplasmic culture. Protoplast are cells without a cell wall but bounded by a cell membrane or plasma membrane. So this is what we are going to culture now. Using protoplast, it is possible to regenerate all plant from a single cell. See what a wonder technology it is. Taking only the protoplast content, we will be able to produce the entire plantlet. Also develop somatic hybrids. The steps involved in protoplasmic culture, the first step would be isolation of the protoplast, fusion of the protoplast, culture of the protoplast, and finally it is selection of somatic hybrid cells. So 4 steps are involved. So let's move into our first topic, isolation of protoplast. As we have learned before, protoplast is the substance which is present inside the cell wall. Without the cell wall, next layer would be plasma membrane. So the content which is present inside the plasma membrane is called the protoplast. So we are going to isolate now. Let's look into the how we can isolate the protoplast. Small bits of plant tissues like leaf tissues are used for isolation of the protoplast. The leaf tissue is immersed in 0.5% macroenzymes and 2% anorexica cellulose enzymes developed in 13% sorbitol or mannitol at pH of 5.4. So what are the materials we need here? 0.5% macroenzymes and 2% Onizuka cellulase enzyme. So then it is dissolved in 13% sorbitol or mannitol and then maintained at pH 5.4. After this, what happens? Let's see. It is then incubated overnight at 25 degrees centigrade. After a gentle teasing of cells, protoplast are obtained and these are transferred to 20% sucrose solution to retain their viability. They are then centrifuged to get pure protoplast as different from debris of cell wall. So this is how we isolate. Now the protoplast is isolated. Let's move into our next step. Fusion of the protoplast. Second step. It is done through the use of suitable fusogen. So fusogen is a new term you are learning for the first time. It is a substance which is used for fusing two different protoplasts. Normally it is PEG. We call it as PEG. What is PEG? Polyethylene glycol. So mark it down, children. Polyethylene glycol is a fusogen which is used to fuse the protoplast of two different cells. The isolated protoplast are incubated in 25 to 30% concentration of PEG, that is polyethylene glycol, with the calcium ions, and the protoplast shows agglutinations, that is a formation of clumps of cells, and then they fuse. So this is how the fusion takes place. So first we isolated using a mannitol, sorbitol, and then soaked with the a pH of 5.4 overnight for 25 degrees Celsius. This is what we have done in isolation. Now this isolated content is soaked, is incubated in 20 to 30% polyethylene glycol with calcium ions for a certain period of time, and the fusion takes place. So this is how the second step takes over. Now we'll move on to the third step. First we discussed about isolation, then the fusion. Now we'll move on to the third step called culture of protoplast. MS liquid medium is used with some modification in droplets, platings or micro drop array techniques. MS medium is nothing but the culture medium, the nutrient medium in which we are going to place the tissue that is need to be grown. Protoplast viability is tested with fluorescent diacetate. This is again a 1 mark question. How can we test the viability? With fluorescent diacetate before the culture. The cultures are incubated in a continuous light 1000 to 2000 lux at 25 degrees Celsius. Again mark it up. So what are the conditions for the culture? They'll be asking. The cell wall formation occurs within 24 to 48 hours, and the first division of cell occurs between 2 to 7 days of the culture. What an interesting thing, no? So the culture takes place. Let's move to the next. After the growth, selection of somatic hybrid takes place. That is our fourth step. The fusion product of protoplasm without nucleus of different cells is called the hybrid. Again another important question, children. What is a hybrid? Hybrid is nothing but the fusion of protoplast without nucleus of different cell. So make note of following this nuclear fusion happens. This is known as the somatic hybridization or hybrid formation. So the entire process has been ended up. Let's revise what happens in the protoplast fusion for one more time. Isolation takes place by using 0.5% microenzymes, 13% 2% onoxical cellulase enzymes, and dissolved in 30% sorbitol and mannitol for a pH of 5.4 and incubated overnight for 25 degrees Celsius. So this is how isolation helps in taking away the protoplasm, and then a fusion takes place. For the fusion we need a fusogenic substance called the polyethylene glycol, and after that the protoplasm culture takes place and the hybrid is formed, resulting in the formation of the hybrid. So now the cells have been grown. In the picture you can find the plant is there, explant is taken. From the explant few tissues are taken, tissues are put into the test tube, and the test tube the protoplast fusion takes place. You can see two types of cells are fused and the process takes place and results in formation of the protoplast culture. Let's move into the fourth type of culture, that is cell suspension culture. The growing of cells, including the culture of single cells or small aggregates of cells in vitro in liquid medium, is known as cell suspension culture. So this is a definition for the cell suspension culture, that is growing of the culture of a single cell or group of cells in vitro, that it is in laboratory or in a liquid medium. So far we have been growing in a culture nutrient medium, no, but now in the liquid medium is known as cell suspension culture. The cell suspension is prepared by transferring a portion of the calyx to the liquid medium and agitated using rotatory shaker instrument. Here comes an instrument for the first time. So we take the substance and we agitate with the rotary shaker instrument. The cells are separated from the calyx tissue and now it is used for the cell suspension culture. So this is how we remove the cell. Okay, we put it in a rotary machine and we shake it up so that the content needed is taken up. Now this content, as usual of our culture procedure, taken into a petri dish and then to the conical flask, then to the test tube where the nutrient is kept and grows into a plantlet. After the full development, transferred as to hardening process to develop into a new plant. So far we have discussed about the various types of culture. Now let's look into the production of secondary metabolite, that is the outcome of the tissue culture. What are the products that we get? Cell suspension culture can be useful for the production of secondary metabolite. So cell culture, that is the fourth type, is mostly used for predicting alkaloids, flavonoids, terpenoids, phenolic compounds and recombinant proteins. So these are the various secondary metabolites. What is secondary metabolite? Make note of it, children. It's another important 2 mark questions. They are chemical compounds that are not required by the plant for normal growth and developed but are produced in the plant as byproduct. We cannot make these substances in any industries or factories. It is a chemical compound only but cannot be manufactured. These are products produced in the plants itself as a byproduct. For example, biosynthesis and isolation of indole alkaloids from Catharanthus rosaceae through the plant tissue culture. So this is how the secondary metabolites are produced. The process of production of secondary metabolite can be scaled and automatized using bioreactors for commercial productions. Many strategies such as biotransformation, eliciting, mobilizations have been used to make cell suspension cultures more efficient in the production of the secondary metabolites. So metabolites are very important. Let's look into the various products which have been manufactured through the plant tissue culture technologies. The secondary metabolites are of very much commercial uses. Let's start. There are 5 important concept products given. Let's see them one by one. Digoxin, it's taken from Digitalis purpurea, and this secondary metabolite is used to cure cardiac. It is used as cardiac tonic for the treatment of the heart. The second one is codeine. It's taken from Papaver somniferum and it is used as analgesic. Third is capsaicin, taken from Capsicum anum. It is used for rheumatic pain treatment. Fourth is vincristine, taken from Catharanthus rosaceae. They have anti-carcinogenic property, that is to treat the cancer cells. The last one is quinine, obtained from Cinchona officinalis, and it is used as treatment for anti-malarial. So malarial treatment, for treating malaria, this is of much value. So all these products are of commercial valued. Let's look into plant regenerative pathway. So for culture we need an explant. The explant may be meristem, shoot apex, stem, leaf, root, calyx. And sometimes you may take directly the embryo also. So embryoid develop into plantlet, plantlets into hardening process and then transfer to the field. In another culture it is indirect embryogenesis, that is we have shoots. The shoots and roots are produced from which embryo plantlets are formed. The plantlets grows into hardening process and then transfer to the plant. The another mode of culture regeneration would be multiple shoot induction, which we also call as a micropropagation, where we have a root induction and from this root we get the plantlets hardening and then the transfer it into the external field. So these are the various pathway for plant to get regenerated to grow into a new plantlets. So we'll discuss of some of these steps now. We are moving into another important topic called somatic embryogenesis. So we are going to produce an embryo. It is a formation of the embryo from calyx tissue directly, and these embryos are called the embryoids, okay, or from them in vitro cells directly from pre-embryonic cells which differentiate into embryoids. So the applications of this embryogenesis is somatic embryogenesis provides potential plantlets which after hardening period can establish into new plant. Somatic embryoids can be used for the production of synthetic seeds. And third is, it is now required in many plants such as Allium sativum, Adnium vulgare, Oryza sativa, Z maize, and this possibly in any plant. So very important thing to be noted is embryo is formed. Second, we call such embryos as the synthetic seeds. Now let's look into what is synthetic seeds. Synthetic seeds are produced by encapsulation of embryoids in agrosol gel or calcium alginate. So we take the embryo and we close them into the substances, that is what mentioned over there, and such seeds are, that is called a synthetic seed. Why did we get this name called synthetic seed? In real seed we got the ovules. These ovules are called their seeds. This ovule have the ability to grow into a new plant, that is the embryo. Similarly here we already have an embryo. So this embryo which is now going to get cultured to many number of embryos, therefore we call such thing as the synthetic seeds. So this embryogenesis is a very important process in modern technology of producing the plants, where embryos are formed from the culture medium. Now let's go into the various Organogenesis. We call it as organogenesis. The morphological changes occurs in the calyx leading to the formation of shoot and root is called the organogenesis. If root is formed, it is called rhizogenesis. If shoot is formed, it is called as calinogenesis. You can see that a calyx is there which is differentiated to form either root or the shoot, and then finally results in the formation of the plantlets. Organogenesis can be induced in vitro by producing plants growth regulators in the MS medium. What are the regulators that we can use? Auxin and cytokinin, which induce shoot and the root formation. So taking up a small cell and making that cell to grow into a new plant. This is called the embryogenesis, and the various formation. Organogenesis is formation of organs that includes both the formation of the shoot and the root called the caliogenesis and the rhizogenesis. For doing this, we need the enzymes, growth regulators called the auxins and the cytokinin. So this is what we have discussed now. Now let's look into the pictorial description of this particular topic. A plant is there. From the plant, explant is taken, and direct somatic embryogenesis is also taking place on one side, where you get a somatic embryo and grows into a plantlet. In indirect method, we take the calyx. The calyx is allowed to grow as a somatic embryo, and from there you get the process going on. And you can also found root organogenesis and shoot organogenesis. Can you tell me what is the name for that? Rhizogenesis and caliogenesis, where the entire plant is formed and it turns to a plantlet and allowed to grow in hardening process and results in the formation of the plant. This is how things. Let's look into the application of plant tissue culture. It has got several applications such as improved hybrid production through somatic hybridization. The second importance would be somatic embryoids can be encapsulated into synthetic seeds, which we call it as synseeds. These encapsulated seeds or synthetic seeds help in conservation of plant biodiversity. The third type would be production of disease-resistant plants through meristems and shoot tip culture. The fourth importance is production of stress-resistant plant such as herbicide-tolerant, heat-tolerant plants. The fifth type would be importance is micropropagation technique to obtain large number of plantlets of both crop and tree species useful in forestry within a short span of time and all through the year. And the final importance is production of secondary metabolites from cell culture utilized in pharmaceutical, cosmetics and food industries. So these are the various applications of plant tissue culture. Hybrids are formed, synseeds, that is artificial seeds, are formed, production of disease resistant and meristem tissue culture is done, stress resistant plants are formed, herbicide tolerance, heat tolerant plants are produced, and micropropagation, prediction of many number of plants in a given lifetime, in a given particular period of time, and finally production of the secondary metabolites. As we have seen the 5 important, not only those 5, apart from that there are many other byproducts that is obtained from the culture. So these are the various applications of the plant tissue culture. Let's look into the types of variation that takes place in an organism. The 2 types of variations are somoclonal variation and gametoclonal variation. Somatic variation found in plants, generated in vitro variations found in leaf, stem, root, tubers or propagule. Whereas gametoclonal variation is gametophytic variations found in plant regenerated in vitro condition, gametic region, that is variations found in gametes and gametophytes. So these are the two types of variations we found in the organisms. One is about the external characters and the other one is with the gamete of the organism. So far we have discussed about the types of plant tissue culture and the direct culture, indirect embryogenesis, the uses. Now we are entering into another area of plant tissue culture called the micropropagation. As I told you before, we are going to take a small piece of the calyx and make it to grow into multiple copies using in forestries in a short period of time all through the year. So this is micropropagation. Now micropropagation of plants at industrial level maintain high standard of homogeneity. They will be having similarity among them in plants like pineapple, banana, strawberry and potato. Here is a protocol given. Let's look into what happens in the micropropagation here. Let's look into what happens in banana protocol. In vitro micropropagation of banana is taken. Sucker surface sterilized with 1% sodium chloride for 30 minutes. Apical meristems are isolated using benzene aminopurine BAP and indolacetic acid IAA. An induction of shoots within 168 days and kinetin 2.0 mg and naphthalene are added for root inductions and accumulation in polyhouses, followed by shade house under 50% light for hardening process, tested for genetic uniformity and transferred to the field. This is how bananas are formed in a commercial scale on a large productions. Next we'll move on to a very important topic called the artificial. Artificial seeds are known as syn seeds, that is synthetic seeds produced by using embryoids, that is somatic embryos without obtained in vitro culture. They may even be derived from single cells from any part of the plant that later divide to form cell mass containing dense cytoplasm, large nucleus, starch grains, proteins and oils, etc. To prepare the artificial seed, different inert materials are used for coating the somatic embryoids like agarose and sodium alginate. So the embryoid is there, the embryo is surrounded by an materials. It may be of agarose or sodium alginate. So this synthetic embryo is formed. Let's look into the application of this artificial seed. Artificial seeds are again known as syn seeds. Artificial seeds have many advantages over the true seeds. First is millions of artificial seeds can be produced at any time at low cost. Production at low cost is the first importance. The second importance is they provide an easy method to produce genetically engineered plants with desirable traits. Third, it is easy to test the genotype of the plants. Fourth, they are potentially stored for a long time under cryopreservation method. Fifth, artificial seeds produce identical plants. And the last point is the period of dormancy of artificial seed is greatly reduced, hence growth is faster with a shortened life cycle. These are the advantages of the artificial seeds. Let's move into virus-free plants. The field-grown plants like perennial crops usually are infected by variety of pathogens like fungi, bacteria, mycoplasma, viruses which causes considerable economic losses. So chemical methods can be used to control fungal and bacterial pathogens but not virus generally. Shoot meristem tip culture is the method to produce virus free plants because the shoot meristem tip is always free from the virus. Let's look into the protocol for this virus free production. Apical meristem tip with 1 or 2 leaf primordia are excised in sterile condition from the explant. In vitro culture in 10 ml of solid MS medium, supplement with growth hormone is taken. Cultures are maintained at 20 degree plus or minus 1 degree Celsius in the darkness for 3 days, followed by normal elimination of 2400 lux. Plantlet developed from meristem tip culture after organogenesis process, transferred to hardening process, and finally transferred to the field condition. So this is how virus-free plants are produced through the culture media. So very much useful for the farmers to get themselves protected from the virus. So meristem culture helps in virus-free production of the plants.